Hello, my name is Emily Elo-Fedrosh, and on behalf of the entire NMDC team, I am delighted to share with you our progress over the past 2.5 years to develop the National Microbiome Data Collaborative. Today, I'm joined by my colleagues Chris Mungle, Leanne McHugh, Patrick Chain, Kirsten Fagnan, and Pajal Van Gay, and over the next 20 minutes, we will showcase how we are supporting efforts towards integrative microbiome infrastructure and inclusive engagement with the research community. Our vision is to empower the research community to harness microbiome data exploration and discovery. And we are founded on the guiding principles that focus on data standards, data quality, data integration, and data access for multi-omics microbiome data. The NMDC aims to deliver a set of unique microbiome data science capabilities aligned with the FAIR data principles, making data findable, accessible, interoperable, and reusable, through leveraging capabilities, expertise, and resources available across our 4 DOE National Lab partners at Berkeley, Los Alamos, Oak Ridge, and Pacific Northwest. The NMDC is supported by 2 strategic priorities: infrastructure and engagement, which underpin the long-term scientific vision to develop and support a collaborative, community-driven data science ecosystem. Our talented and diverse team is pictured here, and we leverage resources at the flagship user facilities, the Joint Genome Institute and the Environmental Molecular Sciences Laboratory, which you just heard about from Douglas and Nigel. I hope that you will take the opportunity to meet with our many members of our team who will be at the NMDC booth, showcasing a poster on the NMDC workflows, or attend our hands-on workshop that will be hosted later today. Last March, we officially launched the NMDC Data Portal and have shared updates and progress on a quarterly basis with the community. You will hear from Chris, Leanne, and Patrick on important updates to our data schema, workflows, and new sample submission system, along with a demonstration of the data portal by Kirsten. And to expand our reach in the microbiome community, we developed and launched the NMDC Ambassador Program, our inclusion, diversity, equity, and accountability strategic plan, among other activities that you will hear more about from Pajal. And with that, I'll hand it off to Chris. Thanks, Chris.
The NMDC is collecting and organizing a diverse range of data about microbiome samples and their environments. This includes their metagenomic data, metatranscriptomic data, as well as proteomic and metabolomic data as well. So we're applying a diverse range of standards here, including MSI, PSI, metabolomics, and proteomic standards, initiative standards, as well as the Genomic Standards Consortium minimal information about any sequence, which some of you may be familiar with. So MICS provides a set of standard checklists and environmental packages, which group together different descriptors that can be used to describe different aspects of a microbiome sample. So NMDC is actively collaborating with the GSC, in a number of different ways. We're working with GSC to engage the microbiome community through the NMDC Ambassador Program. And we're also working to make the standard more modern and machine actionable and make it more fair, that is to say, findable, accessible, interoperable, and reusable. And we're doing this by essentially taking the standard, which was previously available as a spreadsheet, and turning it into something computable using a framework we co-developed called LinkML, Linked Data Modeling Language. This allows us to render GSC MICS as both RDF and JSON schema, and allows us to apply standard data validators to be able to tell if data is conformant to the standard or not. And we are importing MICS as well as other standards such as the environment ontology into our NMDC schema. So all information in NMDC is organized according to the schema. The schema is broken down essentially into 4 different aspects: descriptions of the overall study, any samples collected as part of that study, as well as their different sequencing and environmental characteristics, sample preparation, and so on. Then details of processing of those samples. And finally, the data objects that arise from the different analysis workflows applied to those samples and sequences. And so this schema drives many different things within NMDC. It drives our data submission UI, which you'll hear more about later on. All of the descriptors in the submission UI are driven by the schema. And the schema also is used in our search portal So all of the different terms and fields that you can search on within NMDC are also driven by the schema, and it will also drive our APIs and ways in which you can access the data, and you can hear more about those later.
Thank you, Chris. For researchers to find, retrieve, and reuse microbiome data, the metadata is critical. In addition to the efforts Chris described on metadata standards and organizing that data into a unified schema, we're also developing a portal designed to lower the barrier to submission of standards-compliant metadata. The challenge is that providing complete and accurate metadata is complex and can be confusing for investigators in the field. Our users are not experts in metadata standards or ontologies, and they shouldn't have to be. NMDC's prototype metadata submission interface, shown here on the right, has been developed through collaboration with an open source development project called Data Harmonizer. It supports submission of metadata about a research study, the biosamples, and the multi-omics data being generated for that study. This interface guides the user through the required metadata, and importantly, this tool performs validation to ensure that the entered values follow the correct format and use controlled terms. This submission portal can also track project IDs from EMSL and JGI, and has integrated specific requirements for metadata from both of those user facilities. We're gathering feedback on this portal during our workshop later today. And now I will hand off to Patrick to tell you about NMDC's standardized workflows.
Thanks, Leanne. So in addition to focusing on metadata standards, the NMDC is also focused on standardized and open source bioinformatic workflows. The Nmdc workflows have already had the benefit of being extensively tested within JGI and EMSL for both sequencing data like metagenomics and metatranscriptomics, as well as for mass spec data like metaproteomics, metabolomics, and natural organic matter data. These workflows have been tested on several HPC and cloud environments and are now available for use by the community. The main goal for NMDC workflows and the ensuing analytical products is to allow for more direct comparisons of samples and results across different studies, which promote interoperability and reusability of omics data. Now, internally within NMDC, we've used these workflows to process various projects, including FICUS projects that combine EMSL and JGI data, but we've also extended the ability of our initial workflows to process external data as well. As a pilot, we've partnered with the Earth Microbiome Project to ingest and process some of their raw sequencing data stored in the NCBI sequence read archive. In addition, we've really focused on democratizing these workflows and delivering a variety of options for access by the community. We're working on integrating the NMDC workflows within the KBase environment and have already prototyped the use of the NMDC workflows integrated within the Edge Bioinformatics web-based platform that are hosted independently at the UC San Diego High Performance Computing Center. This option provides the ability for any user to upload their own raw omics data and to utilize the NMDC workflows themselves. And within the app, that provides some additional visualization and interaction with the results of any individual omics data set. Together with documentation on the workflows themselves, we've created a number of additional training material and user guides. And we're always interested to improve both the workflows as well as the presentation of the results. So please contact us if you're interested in becoming a beta tester. We're now turning our attention to workflow orchestration and automation such that jobs can be distributed to available computational resources and so that the workflow results can be automatically uploaded to NMDC to allow researchers access via the NMDC data portal. And so next up is Kirsten Fagnan, who will give us a quick tour of our portal.
Now that we've shared what NMDC has been doing to collect, analyze, and process data, you may be wondering how you can explore and access all of the data that we've put together. So I'm going to spend a few minutes introducing you to the NMDC data portal. This is the view of the main landing page for the NMDC data portal. And there are a couple of tabs here, one that organizes the data by omics type and one by environment. To the left here, there's a search pane that lets you search by all of the facets listed here, as well as a bar for entering text-driven search. There are a number of different ways to view the omics data that are highlighted here. And if you scroll down, you can see the studies associated with the NMDC, as well as a listing of all of the samples. And you can quickly see that there's a lot of data on the NMDC, and we've worked pretty hard to come up with different ways to let users filter this data. A lot of work has been done on the Nmdc schema and data infrastructure to allow users to enter a term and see automatic suggestions for what they might be looking for. If I start to look for Colorado or just type COL, this could be the facet that's the collection date. It could be a geographic location, it could be a collective plant structure, but I can keep typing and I can see that this is the geographic location that I'm interested in. We think this is a really powerful way to help folks navigate all of these different possible search options. I'd like to call your attention to one particular faceted search that we think is pretty powerful and has a lot of potential for making the NMDC an invaluable resource for the community. And that's the ability to search by function across all of the different omics data types. To do that, you can select the KEGG term from the pane, and you can see a dropdown of all the different terms that you might find in an annotation. And if you don't remember exactly what term corresponds to the resource that you're looking for, you can get some suggestions by starting to type in the name of what you're looking for into the search box. So I will select this dehydrogenase here, and you can see that that again filters down the number of data sets and the number of samples associated with that search term. One of the things that makes NMDC unique is how we make it possible for you to find different omics measurements derived from the same sample. And one of the ways we help people do this is through this upset plot navigation. So if you look here, you can see metagenomes, metatranscriptomes, metaproteomes, metabolomes, and natural organic matter captured here along the top. And if you're interested in, say, samples that have a metagenomic component and a metabolome mixed component, you can look down here and see, oh, this also has metaproteomics data. And then you can click on this little bar here and see that there are 33 samples. When you click on that, you can see that the map has updated to show the view of where the samples were taken. You can see the time series information as well, and clearer information about the study as well as the samples. Let's suppose I've decided this is the study I'm interested in, and I'd like to download some of the data associated with the study. And specifically, I would be interested in downloading all of the metagenome assembly files associated with this study. And now I can see that that means that I will get a bunch of different file types, 297 files from all 33 samples, and that the size of this download is 142 gigabytes. Now I need to log in to be able to download this data, so I'll click that button, and I can see now that I'm ready to download this zip file of all of this data. With that, I'm going to turn it over to Pujal to tell you all a bit more about community engagement and user-centered design.
Thanks, Kirsten. So one of the reasons why the NMDC data portal features are so great is because the NMDC team uses a community-centered design approach with all of our user interfaces, that being the NMDC Edge that Patrick showed, the metadata submission portal that Leanne shared, and the data portal that Kirsten just demoed. This means that each time we generate a release, we engage with the research community to review our interfaces and give us feedback for us to consider for the next round of development. This means that the process of reviewing and development is an iterative process, and the step of sitting down with community members is also an involved process that requires us to transform qualitative feedback collected through in-depth interviews with researchers into actionable insights. The best way to see this iterative process is by looking at the evolution of our UIs. So the screenshot on the bottom left corner is one of our earliest versions of our data portal in early 2020. Compared with the current version available on the bottom right corner and a couple of iterations in between. Now, I know that these screenshots are pretty small, so let's dive into this a little bit deeper. So I'd like to share how researchers have directly informed the features on our data portal. Early versions of our search text box were limited in that you couldn't quite search across many terms. Users wanted to be able to search across all terms and all values without having to know ahead of time what the search space looked like or what the organization of that data looked like. This feedback directly informed the latest version of our text search feature that Kirsten demoed earlier, especially that assisted search part. Early versions of our map visualization were not interactive. We observed that users intuitively wanted to interact with the map, and it was consistently the first thing that they noticed. Researchers wanted to select biosamples based on geography by interacting directly with this map. So this feedback informed the pan and zoom feature available in the map that you see today. So in one of our earliest usability interviews, several users noted that they worked with only specific types of omics data, for example, metagenomics and metaproteomics, and it was really important that this data was generated from the same biosamples. But there wasn't an easy way to search for the specific kind of matched omic data. This is how the upset plot, modeled off of a popular R package, came to be part of our interface. And lastly, the ability to choose exactly what you want to download in bulk has been echoed by many of our users and is one of our newer features that we're continuing to refine. So now you might be wondering, who is providing this feedback? Or hopefully you're now wondering, how do I give feedback? Well, let me introduce you to our most engaged community members. First, I want to give a big thanks to them as they've been closely involved in many aspects of the NMDC, which include contributing to user feedback. All of the faces here today represent our champions and ambassadors, which I'll tell you more about in a minute. I do want to clarify that anyone can be involved with usability testing, and you don't necessarily have to be part of these programs, but they do ensure that you're always aware of the latest opportunities. Our Champions program was launched to enable motivated researchers who are passionate about fair microbiome data to engage with us regularly. Champions regularly contribute to usability testing, but they've also beta tested our workflows, reviewed our trainings and documentations, as well as pilot new programs before release. This is a great program to consider joining if you're on the fence about time commitment, but still want to be part of the NMDC. I'm also excited to share that we recently sat down with our champions in a series of listening sessions and have taken their feedback to relaunch the program to better support their needs. This really is a great time to consider joining. Our Ambassadors program is a more formal program that was specifically designed with targeted goals in mind. Our first iteration of the program focused on spreading awareness of metadata standards and understanding how to lower barriers to their adoption. We recruited a cohort of early career researchers across broad microbiome disciplines and worked closely with them to ensure proper training, planning, and coordination so that they could host NMDC workshops and presentations targeting their own communities. Since October of just last year, they've hosted 17 events and have reached about 400 people. We're really excited to share that 2 of our ambassadors are here at GSP and will be presenting tomorrow. So please don't miss their session at 2 p.m. Eastern. In our efforts in reaching the broader community, we've also made a commitment to supporting inclusion, diversity, equity, and accountability, or IDEA, in the microbiome sciences. Last year, we developed an IDEA strategic plan, and we're currently in the planning processes for implementation. We encourage you to take a look at the link provided and get in touch with us if you have any questions, concerns, or contributions. Please note this is a living document, and we hope to evolve it with your input. Lastly, I want to quickly highlight some upcoming events and opportunities. Next week, NMDC team member Alicia Wood-Charlson will be hosting our March community conversation focused on the important topic of data citations. We have a really excellent lineup of speakers from relevant organizations in this space, so we do hope to see you there. And while it's a bit early, we're really excited to share that we'll be hosting a town hall at ASM Microbe this year in June, focused on the important contributions of the research community toward fair microbiome data. Please stay tuned and follow us on Twitter for updates on the date and time. And also, in case you missed it, Microbiome Journal is currently accepting articles for its special collection on experimental multiomics methods that are transforming microbiome research. We highly encourage you to submit your articles as the deadline has not passed. And finally, if you're submitting metadata for a FICUS project soon, please consider piloting our new metadata submission system, where you could submit your own metadata to both user facilities using a single interface. And now I'd like to hand it back off to Emily.
Thanks, Pajao. And with that, we are well on our way to building a collaborative, integrative data science ecosystem for microbiome research. We invite you to visit the data portal, read our latest publications, Engage with our team during this meeting, and particularly join our workshop later today to get hands-on training with the data portal and new sample submission system, and join our efforts to shape the future of microbiome data discovery. Thank you.